Hi, this is Professor Schur. Today we're going to talk about carbohydrates. If you look at the word carbohydrate, it might give you a clue as to what's in it. We see carbo part here, and then the hydrate part, like water, here. So if we look at them, we can talk about carbohydrates as sugars and sugar polymers. And remember, a polymer is a bunch of small units covalently bonded together. We're talking hundreds to thousands of units. And if we look at all of these, they're going to have carbons, hydrogens, and oxygens in them. So that's the carbo, and the H and the O, water, H2O, is the hydrate part. Rarely they'll have nitrogen. So if you're looking at molecule pictures and you're trying to figure out if something's a carbohydrate, you're going to look for carbons, hydrogens, and a decent amount of oxygens, as well as carbonyl groups. Remember, those are the C double bonded to the O, and hydroxyl groups, which are the OH groups. We have three different categories of carbohydrates we'll talk about. There are the single sugars, so they're a single ring. We call them monosaccharides, mono meaning one, saccharide meaning sugar. And they're the monomers of carbohydrates. Then you can take two of those monomers and you covalently bond them together and you make a disaccharide, di meaning two, saccharide, sugar. So it's a double sugar. Then you take hundreds to thousands of these monosaccharides and bond them all together with covalent bonds, and you get what we call a polysaccharide. Poly means many, so that's many sugars. As far as monosaccharides go, you're going to want to know the structure and function. Structure you can read about in your textbook and on Mastering Biology. Whenever you're seeing words with the ose ending, like sucrose or glucose or fructose, usually that means sugar, and it will almost always, to my knowledge, mean carbohydrate at least. So if we look at these, we have twice as many hydrogens to carbons and oxygens. And I should mention this CH2O, like a multiple of that, is only for monosaccharides. Because when you start putting monosaccharides together, you do that through dehydration synthesis, and if you recall, you're pulling out of water every time. So you're going to lose two H's and an O each bond. So keep that in mind. But you can see here glucose C6H12O6, that is a monosaccharide. And basically you're going to look for their carbonyl group. I can show you here. So here's some carbonyls. Remember, the C double bonded to the O on the end is called an aldehyde, and so this is an aldose. The C double bonded to the O in the middle is the ketone, so this is a ketose. There's different length backbones that carbohydrates can have, all these monosaccharides. And ones that are longer than 3 tend to form rings when they're in aqueous solution, so when they're in water. And as you know, the body is made of predominantly water, 70 to 95% water in a cell. And so this is the stage or the state that they're going to be found in. So if we actually look at the ringing up of the linear molecule, and it does fluctuate back and forth, but it's preferentially the water form, it's preferentially the ring form in water. What's going to happen is that we have this OH here, and we have the carbonyl group here, and they're going to ring up, and you're going to actually form a bond between the oxygen and then the carbon. And so you get some rearrangement. The hydrogen doesn't actually jump straight over here, but you get some rearrangement, and then you formed a bond. So you can see, when you see a ring with an oxygen in it, that's going to be a good hint that that's a carbohydrate, a monosaccharide, or monosaccharides built into something larger. And that's the remnants of the hydroxyl group and the carbonyl group joining. So you'd be looking for something like this. It might not have all the lines off of it. Sometimes we don't draw it with those. But the oxygen in the ring is telling you carbohydrate. And so here's some examples of two C6H12O6 monosaccharides. One is called glucose. The other is called galactose. And you can see it's very similar. They're isomers. The only difference is the position of this hydroxyl group. So when the galactose rings up, it actually has this hydroxyl group on this side of the ring. And once it's in a ring, it's stuck in that conformation. Glucose has this hydroxyl group on the bottom side of the ring. And these make them different enough to where They are recognized only by particular enzymes to be worked on. They only trigger certain responses in cells, and they only build certain molecules. I want to go back to the functions of monosaccharides. Really, there's two of them. So first of all is fuel for the cells. And when we say fuel, we actually mean a source of potential energy. So the bonds, all these carbon-hydrogen bonds, are going to be energy containing, meaning that we can break down glucose in particular and extract energy from it in order to make ATP, a molecule that allows the body to work, all the cells to do the reactions and pump things across their membranes and muscles to move and all that kind of stuff. So that's one thing, fuel for the cells. And secondarily, monosaccharides can be broken down into their almost atomic level or very small molecule level and then rebuilt into other small molecules. So let's say there's a lack of a certain type of protein building block called an amino acid in your body, but you have glucose coming in. Frequently you can rearrange glucose and just combine it with some other stuff and send it out as that building block for the protein. Now, not all the time. Sometimes you have to eat certain building blocks, particularly for proteins. But this rearrangement, that's what raw material means. Then we get to disaccharides, the two. There's still OS, and both monosaccharides and disaccharides will taste sweet if you taste them. So you can see these are just two monosaccharides covalently bound together, and dehydration synthesis is how that occurs. So dehydrate means to take out a water, if you recall. So here's a hydroxyl group, here's a hydrogen of another hydroxyl group. They come together and take out the water. We have two free bonds that are not satisfied with their electrons, and so those two guys come together. And then we have a bond between the two monosaccharides. It's called a glycosidic linkage. You'll notice there's an oxygen in there. So if you're looking at molecules and you see ones with O's and rings and oxygens between the rings, that's a good sign that that's a carbohydrate. And if it's just two, that would be a disaccharide. You can have all kinds of combinations of monosaccharides. The book talks about glucose, fructose, and galactose combinations. So maltose, which you use for brewing beer, it's malt sugar. Sucrose, which is table sugar, and also is what plants move around their body. So after plants make sugar, make food, they'll produce glucose, which will then get rearranged, one of them to fructose, and then gets put together, and the sucrose is what travels throughout the plant. There's also lactose, which is the milk sugar. So lactose is a glucose and a galactose monomer together. There are lots more disaccharides. Those are just the three that happen to be mentioned in our book. And then we get to the many sugars. So these really we don't really call them sugars anymore. We call them sugar polymers, but the most proper name would be polysaccharides. These are very, very long. They're macromolecules, and there are two main functions of polysaccharides. Now, these aren't going to be sweet anymore. At least, yeah, they're not going to be sweet when they're in their full form. These are going to be like your starches are part of this group, so kind of what you think of probably as carbohydrates in your diet. A lot of them are polysaccharides. So they are energy storing as well as structural components. And energy storing makes sense because if you think about what a polysaccharide is, it's a big long molecule of monosaccharides. And we just said monosaccharides are energy sources. So if you have a big long polymer of monosaccharides, you're storing energy. You will want to know which polysaccharide does which kind of function in animals or plants and other organisms. So there's a chart you can fill out to try to help you organize those thoughts. You can of course make your own stuff as well. So for example, in plants, the storage polysaccharide is called starch. You're familiar with that from potatoes, whole grains, things like that. So it's a bunch of glucose monomers. They store them in membrane-bound sacs in the cell called plastids, And both plants use their own starch for food, and animals use starch by eating plants for food. But animals don't produce starch. It's just the plants. Animals, on the other hand, when they need to store carbohydrates or monosaccharides, they're going to store them in the form of glycogen. So glycogen is pretty similar to starch, but it's more branched. Also made of glucose, and that's going to be in your liver and muscles. So when you are using your first stores of energy, let's say you go out running, that's going to be your glycogen. That's what goes first. As you can see here, here's the starch. It's a helix, single chain. You can see oxygens in the rings, oxygens between bonds. Here's your glycogen. You can see that there's branching parts off of it. And so that's going to be they have the muscle here. So that's the storage in the muscle. The starch is the storage in the potato. And then we have our structural polysaccharides. So one of the structural polysaccharides, giving strength to structures in either the cell or in an organism as a whole, this one is called cellulose. I'll come back to that picture in a minute. So cellulose is in plants. It is the most abundant molecule because there are so many plants on the earth. And it's going to make the cell walls of plants. So a plant cell has a cell membrane, and surrounding that is this tough cell wall, which makes plants sturdy, gives them their structure to a large degree. And this cellulose is made of starch monomers as well. So you're thinking, well, everything is made of, or excuse me, this cellulose is made of glucose monomers. So that's the same as starch was made of glucose monomers, glycogen's made of glucose monomers. So how are these all different? We said the branching is different for starch and glycogen. And in this case, cellulose, there's two different forms of glucose. And all it depends on is when the glucose forms the ring, if the OH is trapped on the bottom or the top of the ring. And you remember, these are flat representations, but all of this has three-dimensional structure. So when the glucose is trapped on the bottom, if those get put together, they make starch. And it turns into a helix. Good for storage. If they are on the top of the ring, the hydroxyl group, then the only way that the monomers can go together is by alternating upside down and right side up, which doesn't allow this helix to form. And that's the beta glucose. So you can see here, instead of the helix, our cellulose is long, straight molecules. And actually they have groups, hydroxyl groups, that can hydrogen bond. So they hydrogen bond between each other, and there's many, many individual strands, and they bundle again and make very strong material, which is what helps the plants make their cell walls. Finally, we have let me mention one more thing about cellulose. So when we say insoluble fiber on the back of a package, most of what we're talking about with insoluble fiber is cellulose, because this shape can be broken down by human bodies and most other animals, because they have the right shaped enzyme, the protein, that can break apart the bonds. Cellulose, on the other hand, most animals cannot digest cellulose. So they don't have the right shape to tackle this sort of structure. So they can't break the bonds. So insoluble fiber is predominantly cellulose. There are some animals or organisms that can break it down. So termites eat wood, which is cellulose, but termites can't actually break it down themselves. In their gut, they have this protist called trichonympha, and trichonympha has the right molecules to break down the cellulose. It can give some of those to the termite, and then the termite can actually eat wood for energy. Because in order to get energy out of a polysaccharide, you have to break it into its monosaccharides first. That's how it gets fed into the system. Cows and deer, we call them ruminants, they're all going to also have bacteria or protists in their guts that help digest the cellulose and then can give it to the ruminant to eat or use for energy. And then finally, our last type of carbohydrate is polysaccharide. It's a structural one, and it's called chitin. I know it looks like chitin, but it's not. It's called chitin. And chitin has nitrogens on them. This is one of the limited carbohydrates you'll see with nitrogen. But you still see it has the ring with the oxygen in it. It has the hydroxyl groups. There's a carbonyl group in here before the ring formed. And this one is going to be also very strong. So these kinds of appendages here, the nitrogen group, can actually hydrogen bond to each other and make this strong sheet. So that strong sheet can be used for exoskeletons of arthropods, which are like insects and spiders and crabs and crayfish and all that stuff. So their external skeleton is chitin, which is actually sort of leathery before it gets calcified. So chitin itself is not brittle like you might think of a cockroach shell as, like crunching. So the crunching is the calcified chitin. But if you ever see like right after molting, if you see an insect, it's kind of leathery, or like soft-shelled crab, that's its chitin before calcification. It's also found in fungi and algae cell walls. So they also have cell walls. Plants are cellulose. Fungi and some algae can be chitin. And then also we use it in surgical thread. So some surgical thread's made of chitin, which is strong, but it dissolves in the body, and so then you don't have to take your stitches out. So that's what I have for you about carbohydrates. Like I said, you should look at the table, and that's going to help you organize your ideas, make some concept maps, that kind of thing.